Hello, Engineer Tamboli sir. Can you hear me?
Yes, yes.
Okay. A very good morning to one and all present here for this 2 days webinar on advances in basic plant research in relation to climate change. I have 3 important announcements before we start the session. The participants who have not joined by their name are requested to rename themselves because it is important for recording your attendance during the session and also to recognize the registered participant. Secondly, if you have any query or questions related to topic during or at the end of the lecture, you can write those in the chat box. We shall open the chat box toward the end of the lecture. And please register yourself from posting unwanted messages. And thirdly, we shall post one short assignment on a WhatsApp group at evening, and all participants are requested to submit the assignment on time and as per the guidelines issued on WhatsApp group. And please note that responding to this assignment will be considered for issuing the certificates to the participants. So friends, we are in the second day and this is last day of this webinar. And today we are having 2 sessions. And in first session, we have with us Dr. Ranjan Kumar, a Senior Scientist, Division of Biochemistry, IARI, New Delhi. So on the behalf of CAST project and Department of Biochemistry, Mahatma Phule Krishi Vidyapith Rahavli, I extend warm welcome to you, sir. I also extend warm welcome to Dr. R.M. Naik sir, Head Department of Biochemistry and Organizing Secretary of this webinar. And without wasting much more time, I would like to request Dr. R.M. Naik sir, Organizing Secretary of this webinar, to introduce Dr. Ranjan Kumar sir.
Hello, good morning one and all. It's a pleasure and privilege to say a few words regarding the learned speaker today, a very young and dynamic researcher, Dr. Ranjan Kumar, who is a senior scientist at Division of Biochemistry, IARI, New Delhi. He joined the ICR services in the year 2008 and has done a commendable work in the area of stress proteogenomics particularly, and he has published many papers in international, national journal of peer-reviewed journals. He has been a coordinator of CAFT programs, that is Center for Advanced Faculty Training, and 5 such programs he was coordinator. He has got 9 ongoing projects funded by different funding agencies. He has completed 5 projects up till now, and he has prepared training manuals, around 8 training manuals. Dr. Ranjan did his MSc from Acharya Narendra Dev University of Agriculture and Technology, and he did his PhD from IARI, New Delhi. And he joined the services in the year 2008 as scientist in IARI Division of Biochemistry. As I mentioned that he has guided many students, MSc and PhD students, and particularly the noteworthy findings of his research over the years is he has identified and validated 4 novel heat responsive SSR markers, which will be useful for the development of climate smart through breeding program. Dr. Ranjan has also developed thermostable RuBisCO Activase enzyme from wheat through protein engineering, that is site-directed mutagenesis, and the enzyme can tolerate temperature up to 45 degrees centigrade for 1 hour without losing its activity. Dr. Ranjan has 37 novel miRNA, microRNA, has been identified from wheat, and 5 were validated to be heat responsive, reported in wheat. These novel microRNA, miRNA, can be used in wheat breeding program for the development of thermal-tolerant variety. She will be talking today on a topic, the unsung hero of carbon assimilatory pathway, unfolding the story of RuBisCO Activase in which under terminal heat stress through Omics approach. As you know that heat stress will be a major threat to crop productivity, and in that direction, this is a very pertinent topic. And what Dr. Ranjan has done, and he has he will be sharing with us. Dr. Ranjan has got many awards and recognition to its credit. NASI Associateship Award, Young Scientist Award, Young Scientist Associate Award, Scientist of the Year Award, and many more such awards he will be getting because he's doing a wonderful work in this focused area of heat tolerance. So without wasting much time, I request Dr. Ranjan Kumar to present his topic. And I welcome all the participants for this today for this particular lecture. With these words, I welcome again you, Dr. Ranjan, and I request you to proceed further with your presentation.
Thank you, sir. Just give me 5 minutes to share my slides.
Yeah.
So is it visible?
Yes, sir.
Okay. Slide.
Yes.
Hello.
Yes, sir.
Yeah. Thank you, Naik sir, for using such a kind word for me and giving me opportunity to share my work with you people. And thank you to NHP CAST for
Hello, Dr. Ranjit sir, please unmute.
Hello.
Okay, sir, fine.
Oh, sorry, because it was muted. So again, I'll start. Thank you, Naik sir, for using such a kind word. And I welcome all the participants who have joined in this webinar. So maybe in a couple of minutes, I'm going to unfold the story of two very important friend or foe, that is RuBisCO and RuBisCO Activase. So these are the key players in carbon assimilatory pathway. So maybe I'll be sharing some of the achievements which we did, and one RuBisCO Activase, thermostable RuBisCO Activase, which we developed. So the title, if you say, the title says Unsung Hero of Carbon Assimilatory Pathway, unfolding the story of RuBisCO Activase in wheat under terminal heat through Omics Approaches. So here maybe I'll be sharing some of the omics which we did to identify the RuBisCO as well as RuBisCO Activase in one of the staple food grain crop, that is wheat, and how we use different tools to develop a thermostable RuBisCO Activase enzyme. So before starting, I think let's have a few slides on one of the major problems which we all are facing, that is global climate change. So if you see the forecast of different organizations, international organizations, so it has already been forecasted that we are expecting 0.5 to 1 degree Celsius increase in temperature maybe in coming years. If you see the year 2016 and very recently 2019 has been forecasted as one of the warmest years. If you see other parameters, so there is a long trend of rising global temperature, and the effect is very much visible in the production, productivity, as well as the quality which we are going to harvest. So recently, a report has been published by UNO where they claim that because of this increase in temperature, the quality of the grain has depleted, or most of the beneficial nutrients that has been depleted from the grain. So this is a grave concern for all of us, because even if we increase the yield, so ultimately what we require is a grain with a nutrition. Ultimately what we require is a nutritious grain with a quality. So global climate change, earlier it was forecasted as a myth, but now truth is that it is a reality and we all are a part of this climate change. If you see some of the data which has been released, like UNO, they have forecasted 3 to 5 degrees Celsius rise in temperature, maybe by 2100. And if you see the years, so 1998, as I told, 2016 and 19 has been forecasted as a warmest year. And for next 2 decades, we are expecting almost an increase of 0.2 degrees Celsius per decade. If you see by the production, or if you see the effect of this increase in temperature on production, so very IARI itself, so they have done a lot of work, field-based work, and it was forecasted that there is a decrease of almost 4% in the yield for every 1 degree Celsius rise in temperature. So if this is the case, so definitely it will be difficult to feed the burgeoning population in years to come. So coming on to the abiotic stress. So basically, as I told, my expertise is in stress proteogenomics, especially I worked on wheat. So if you see the abiotic stress, so this is now one of the major concern for all of us. So it's not only decreased the yield of most of the agriculturally important crops, but now we are also compromising on the quality front also. So different omics tool has been used in the past to develop climate smart crops, but then we have very limited success in this area. If you see the wheat, so you all might be knowing that wheat is one of the staple food grain crop all over the world, which provides almost 12% of the calorie and 40% of the energy in our diet. So this is the importance of wheat. And if you see the effect of climate, so almost 6% decrease in yield has been forecasted for every 1 degree Celsius rise in temperature. So we all know the critical stages of wheat and terminal heat. As I told for students, let me tell you this terminal heat, basically terminal is the end. So any increase or fluctuation in temperature during the grain filling or during the end of life cycle of the plant, so that has drastic effect on the quality as well as the quantity of the grain. And this is called as terminal heat. So if you see the field data, so almost it has been reported that there is a decrease of 300 kg per hectare for every 1 degree Celsius rise in temperature for a week. So even it has a drastic effect on the source and sink efficiency. The ratio is highly disturbed, even if there is a fluctuation of 1 or 2 degrees Celsius in the temperature. Even we have seen that even a fluctuation of 1 degree Celsius temperature during pollination and grain filling stage has a severe effect on the synthesis of a starch granule in case of wheat. So now photosynthesis, which is one of the major constants under heat stress. So even if there is a fluctuation of 1 or 2 degrees Celsius, so it has been reported that there is drastic decrease in the photosynthetic rate. Now question comes, so why there is a decrease in the photosynthetic rate? So it's basically because most of the key enzymes or metabolic pathway associated enzymes, so they are heat labile. So even if there is a fluctuation or increase or elevation in the temperature by 1 or 2 degrees, so that has a drastic effect on the activity of these key enzymes associated with photosynthesis or carbon assimilation. So if enzyme is not able to act in the pace as designed, so it will have an effect on the final product. So reaction will not move with the efficiency. It's not only the enzyme. So even it has been reported that if temperature goes beyond the ambient, like 38 or 40 degrees Celsius, so it generally denaturates the enzyme. So once enzyme is denaturated, so you cannot expect the reaction to move with the same pace. Similarly, if you see the effect on the CO2, carbon dioxide, so people, they have reported that with the increase in the temperature, so there is a lower CO2 solubility, which limits the carbonylation reaction, and ultimately it declines the photosynthesis. Even with the increase in temperature, so people, they have reported increase in the photorespiration rate, which is unnecessarily lost on the carbon assimilation. If you see the ROS production, so with the heat stress, this is well accepted that there will be oxidative rust. A lot of reactive oxygen species as well as reactive nitrogen species will be formed. And these reactive free radicals are very dangerous, not only to the organism, but also to the redox system of the cell itself. So it has an imperative effect on different metabolic reactions or different pathways which is operating inside the cell. And the effect is very visible in terms of the outcome or product of the reaction. If you see the leaf structure and integrity, so that is also compromised when a plant is exposed to heat stress. Even some of the electron transport chains and other reactions which is going on, it is highly disturbed because of enhance in the temperature, which obstructs the ATP synthesis also, and then ultimately the effect is visible in terms of decline in the photosynthesis rate. Now in biochemical term, if I have to explain the effect of heat stress on photosynthesis, so here if you see almost all the key enzymes which we know are involved in photosynthesis like RuBisCO or RuBisCO Activase, or maybe the chlorophyll pigment, so it disintegrates with the increase in the temperature. Even if you see the lipid peroxidation, especially membrane, the ratio of saturated and unsaturated fatty acids, so that is highly disturbed when there is an increase in the temperature. Even the activity of some of the enzymes which are involved in transportation, that is disturbed, because of which the transportation of photosynthesis from leaf to the stem via to the endosperm is disturbed. If you see the grain weight and yield, so it is highly affected even if there is an increase of a few degrees Celsius temperature during a grain filling stage, especially milky ripe stage. So it leads to decrease in some of the key enzymes which are associated with the starch biosynthesis pathway, like ADP glucopyrophosphorylase or soluble starch synthase or starch branching enzyme. And it's not only that it will have an effect on the starch granule synthesis, but even the composition of starch, especially the ratio of amylose to amylose pectin is highly disturbed because of this increase in the temperature. So that ratio is not maintained. Even if you see the grain, or if you see the scanning electron microscopy of this grain, so a lot of empty pockets are formed because of this disturbance in the activity of enzyme and because of formation of a small and fragmented granules, which is visible in terms of a shriveled grain formation. So it's not only the effect of heat stress which is visible by eyes, but it has an internal effect also which is visible in terms of quality when you use that product. So these are some of the effects. This is one work which we did in our lab. Here you can see the effect of heat stress during pollination and grain peeling. So first, if you see, this is the pollen of wheat under control and heat stress condition. So under heat stress condition, when during pollination stage, when there is an increase of 2 to 3 degrees Celsius temperature, so it leads to formation of flaccid pollen, or pollen will burst out, and then the pores or the rhizae which is present on the surface of the pollen, that will be clogged with this starch. So this clogged means pollen is not viable, and when it fall on the stigma, it will not able to grow and form the pollen tube. Similarly, if you see the stigma, so under control condition, it is very fine, well-ribboned stigma. But when there is an increase of temperature or when a plant is exposed to heat stress, so this well-fine ligand stigma, so it will unfold, all these lines will come out, and then when a pollen comes and sit on this, it will not able to get the nourishment which is required for the growth of the pollen tube, and ultimately pollen will not able to grow. If you see the starch, the effect of heat stress on starch, so this is one of the SEM of beet cultivar SD2985. So here you can see that under control condition, we have a very beautiful globular starch, and when you make chapati out of this, so even seeds will have a very good shape, and then when you make chapati out of this, so it will have a taste. But when a plant is exposed to heat stress, so here you can see the pleated starch granule. It is a small pleated one and glued to each other. So this leads to formation of empty pockets, and the effect is visible in terms of shriveled grain formation. And when you make chapati out of it, that taste will be not palatable. It will be not good. So these are some of the effects of heat stress. Now coming on to the today's topic hero, that is RuBisCO Activase. So let me introduce you to the RuBisCO Activase. So RuBisCO Activase is a catalytic chaperone. So this RuBisCO Activase basically activates the inactive form of RuBisCO. You all might be knowing that RuBisCO generally presents in the leaves in inactive form, and it requires a catalytic chaperones to convert itself into the active form for carbon fixation. And RuBisCO Activase is the catalytic chaperone or friend of RuBisCO, which helps the conversion of inactive form of RuBisCO into active form. But the problem is that this RuBisCO Activase has not much been characterized in most of the agricultural important crops. Let me tell you, when we started our work in wheat, so that time only 2 partial fragments of RuBisCO Activase gene was present on NCBI dome. So much information was not available, and few crops, few model plants, so they characterized, and then some of the informations were known about RuBisCO Activase. So this RuBisCO Activase, as I told, this is a nuclear encoded cytosol synthesized protein, and it belongs to the AA+ family of ATPases. So it catalyzes both the RuBisCO activation as well as ATP hydrolysis. So all short of conversion from inactive form of RuBisCO to active form it used to take. The only problem is that RuBisCO Activase is highly heat labile. Highly heat labile means it has an ambient temperature of 32 to 33 degrees Celsius, or even if there is a fluctuation of 1 or 2 degrees Celsius, so it has a drastic effect on the activity of RuBisCO Activase. So just foresee a situation where there is an increase of temperature up to 35 degrees Celsius. So in that case, so RuBisCO Activase, it will not able to show its activity as expected, and because of this, the inactive form of RuBisCO will be more as compared to the active form, and that is visible in terms of decrease in the activity of or decrease in the photosynthetic rate. If you see the genes, as I told, very few genes has been characterized and reported and is present on the public domain when we took this experiment, and then we identified a lot of genes from wheat. So maybe in coming slides I'll show you, and then how we planned and identified that story also I'm going to share with you. So as I told, because of its heat labile nature, so it has a drastic effect on the activity of its friend, that is RuBisCO, which is visible in terms of decrease in the photosynthetic rate. Coming on to the reaction. So as I told, so this is the common reaction which takes place inside the leaves. So you have a RuBisCO inactive, which requires a catalytic chaperone, that is RuBisCO Activase, in order to fix the CO2, and this happens under normal conditions. So this is the RuBisCO Activase, which basically removes some of the sugar phosphate moieties, which basically go and sit at the active site of this RuBisCO to make it inactive. So RuBisCO Activase, basically it removed all these inhibitors or phosphate moieties from the active site, and in turn convert this inactive form of RuBisCO into active form. But when heat stress happens, or when a temperature rose above 35 or 36 degrees Celsius, so this RuBisCO Activase is not able to work, and in that case, the active site of this RuBisCO is already inhibited by these sugar moieties or sugar phosphate moieties, and then because of this, RuBisCO will not able to fix the carbon dioxide, and ultimately you can see a drastic decrease in the photosynthetic rate. So this was the mechanism which we thought, and then we planned that let us remodel this RuBisCO Activase, so make it thermostable, so that at least this decrease in the photosynthetic rate, which happens because of increase in the temperature, so at least that will be robust, and then the percent decrease in the photosynthetic rate, so that will be also equalized. Let me tell you, it has been reported that if somehow we get success to get a very efficient thermostable RuBisCO Activase, we can increase the production by 10%. So this is forecasted. Here, if you see the RuBisCO, a real victim of heat stress. So you all might be knowing that RuBisCO, basically, this is the key enzyme involved in the carbon fixation. And as I told, so this is present in the inactive form. Now this RuBisCO is prone to form a different misfired products. And these products are a real problem for RuBisCO, because these products will go, it will sit onto the catalytic active site, and then it will hinder the reaction to execute. So with the increase in temperature, so CO2 diffusibility that will increase, there is an increase in the photorespiration, and there is a decrease in the activity of RuBisCO Activase, and all these clubbed together leads to decrease in the carbon fixation or photosynthetic rate of the plant, and ultimately that is visible in terms of decrease in the yield as well as the quality of the grain which we are going to harvest. So this was the basic information which we had. Now, gap in the RuBisCO and Activase. So some of the gaps which was there at that time, like very few RuBisCO and RuBisCO Activase genes were reported from wheat. And here I've used the term wheat, but then even in other agriculturally important crops also, very limited information about the genes were available on public domain. Even the protein structure, so protein of this RuBisCO and RuBisCO Activase were only characterized in model plants. So it was not much characterized in like cereals or some other important grains. Even the interaction between these two proteins. So this was known that RuBisCO Activase acts as a catalytic chaperone, and then it converts inactive RuBisCO into active one. But then the interaction between these two proteins, that was not known. Even the regulation of RuBisCO and RuBisCO Activase, that was not explored. even the effect of this RuBisCO Activase on yield, because what I told, it was forecasted, it was predicted through simulation models, but then actual effect, that was not known. And the biggest problem is that till date, we don't have a standard protocol to analyze the activity of RuBisCO as well as RuBisCO Activase, because the method which is available, that is radioactive labeling based method, which is not feasible, or which is not feasible to execute at every place. So this is one of the problem and gap in RuBisCO and RuBisCO Activase research. So let us share some of the achievements or our story, that how we started and then how we achieved some significant results. So first we started with the screening of different diverse genotype of wheat. So we have a core set with us which has been developed for thermotolerance, where diverse wheat genotypes were there. So we started screening these diverse wheat genotypes for RuBisCO as well as RuBisCO Activase activity. So here you can see the RuBisCO Activase activity in diverse genotype of wheat. And then we could able to make out that some of the genotypes like RAS 3765, WR544, SD2781, HALNA, and then HI1531. So they really performed very well even under the stress condition. And the activity was quite high as compared to the other genotypes. Here you can see all blue histograms. So all blue one are thermosusceptible, and then yellow one are thermotolerant wheat genotypes. So once we screened this RuBisCO Activase, and then we got some of the lines which showed very high activity of RuBisCO. So we also analyzed the activity of RuBisCO. So here you can see the activity of RuBisCO in diverse genotype of wheat. So based on the activity, we could able to identify some of the genotypes like RAS 3765, SD2888, C306, HALNA, and then SD2932. So these were some of the genotypes having very high activity of RuBisCO as compared to other genotypes. And we could also able to conclude that the activity of RuBisCO as well as RuBisCO Activase was very high in case of thermotolerant as compared to thermosusceptible, except few cases where we got high activity in a thermosusceptible cultivar also. So this was one of the conclusive outcome of this work which we did. And then we could able to identify some genotypes also, which we plan that in further characterization, we'll use those genotypes. So once we did this activity assay, which let me tell you is not easy, as I told earlier, it is a radioactive labeling based method, and you have to devote ample of time to characterize, to go for activity assay analysis. But then our objective was to find out the RuBisCO Activase which is present in wheat. So first we started with the tool which is very known to all of us, that is gel-free proteomics. And then we used this nano LC-MS, which is one of the tool of gel-free proteomics. And then we used one crop also along with the wheat, that was maize. And we could able to identify a couple of differentially expressed proteins. And we expect that definitely one or the other RuBisCO Activase will come in this differentially expressed protein. But to our agony, we got only one RuBisCO Activase along with other stress-associated proteins, which we identified through this Orbitrap. So here you can see through Orbitrap, we could able to identify some of the unique peptides in control as well as heat shock treated. And we also identified some of the differentially expressed stress-associated proteins, and then RuBisCO Activase. So one RuBisCO Activase protein we could able to identify. But then we started characterizing this through reverse approach to clone the respective gene, but then it was with limited success. Then further, we planned to use other higher end of this proteomic tool, that was iTrack. This is basically isobaric tag for relative and absolute quantitation of protein. So use this iTrack to identify some of these differentially expressed protein in contrasting wheat genotype. So here I am just presenting a result of one genotype, but we did in almost 8 different genotypes of wheat. And we could able to identify almost 5,500 proteins with high accuracy. And out of this, we could able to identify 3 RuBisCO Activase from the wheat. And this was first time that 3 different RuBisCO Activase we could able to identify using this iTrack. And then we used that peptide sequence to design the degenerate primer. And then we started cloning that. So we got success in cloning one, which was cloned. Then we could able to make it full length through RACE. And then that sequence was submitted in NCBI. So this was our first success. But then further, we thought that there is some problem with the protein. Let us start with the transcript itself. So we plan to go for a substantial subtractive hybridization. So we constructed different SSH library using contrasting wheat genotypes, and then we started characterizing this. But then we could not able to find out any RuBisCO Activase out of this SSH library. So that was little bit setback for us. So we got almost different differentially expressed genes or ESTs. Almost more than 500 ESTs were identified. But then the problem was we could not able to identify any RuBisCO Activase. And if you see the post-translational modification characterization, so here also we could able to prove that under heat stress, it is the phosphorylation, which is phosphorylation frequency, which is the maximum as compared to other translational modification. So this was one outcome, but then our objective was to find out the RuBisCO Activase. So that was somehow not feasible with SSH library. Then we just moved one step ahead and we thought, let us go for de novo transcriptome sequencing. So we planned experiment with few contrasting wheat genotypes like SD2985, SD2329, RAS3765, and BT Schomburg. And then we did transcriptome profiling. And here you can see we could able to identify almost 80,000 stress-associated genes. Almost 1,200 microRNAs were identified through one of our other experiments. And out of that, we could able to prove that 42 are heat responsive. So like this, we characterize, and then based on characterization, we could able to predict, we could able to identify 12 novel RuBisCO Activase from wheat. And these were not duplicated sequence. These were unique sequence, which shows homology with the domain which is present in RuBisCO Activase. So we predicted 12 transcripts showing homology with RuBisCO Activase, and then we started characterizing and cloning these transcripts. So here you can see we could able to clone 5 different RuBisCO full-length RuBisCO Activase genes. The sequence was characterized and submitted in NCBI GenBank. It's not only that using the information which we got, we could able to clone from wheat, but same we designed degenerate primer, and we could able to clone almost 4 novel RuBisCO Activase from maize also. So that was characterized and submitted in NCBI GenBank. Now see the story. We started with empty-handed, but because of these advanced tools, so we could able to identify 12 novel RuBisCO Activase, and out of this, we got success in cloning almost 5, and then that sequence was characterized and submitted in NCBI GenBank. So this was these are some of the amplifications and cloning results which we got. So it's not only that we cloned and submitted in NCBI, we started doing characterization. So here you can see this is a domain scanning or domain map of a RuBisCO Activase cloned from a thermotolerant and thermosusceptible cultivar. And we could able to find out variations in the amino acid sequence also. So there was a lot of patches which differs in the same gene cloned from thermotolerant as well as thermosusceptible cultivar. So this gives us some idea that there is some significant variations, or there are some significant sites which decides the stability of this RuBisCO Activase. So this gives us an idea that definitely if we manipulate or if we mimic the RuBisCO Activase, which basically is present in some of the hardy crops, so it will be not difficult to design and develop a RuBisCO Activase protein, which will have a very high stability against heat stress. So that was our hypothesis, and we started venturing in different directions. As I told that once we cloned the gene, so we started characterizing this RuBisCO Activase through real-time PCR. So here you can see we collected samples at different stages under differential heat stress in different tissues, and then we could able to characterize the expression as well as the activity of RuBisCO Activase. And from this, at least we could able to conclude that it is the milky or the milli-ripe stage where RuBisCO Activase activity is very high. And it's not only the activity, even the expression of RuBisCO Activase is very high during the grain filling stage. And in grain filling, especially the milli-ripe stage, the activity is very high. So that was the outcome or conclusion out of this experiment. Then further, we characterize the RuBisCO, because whatever you do with RuBisCO Activase, your experiment or your result will have no use unless you characterize the RuBisCO, which is the main enzyme of your carbon fixation. So we started doing expression for profiling of RuBisCO Activase as well as RuBisCO under different stages, under differential heat stress, and here you can see the result, the expression of RuBisCO Activase under differential heat stress, and then the expression of RuBisCO large subunit under differential heat stress. And we could able to conclude that RuBisCO large subunit, it shows down regulation at a temperature of approx 38 degrees Celsius for 2 hours. And then this was observed to be more heat labile as compared to the RuBisCO Activase in both the contrasting heat genotypes, that is RAS3765, which is a thermotolerant, and then SD2329, which is a thermosusceptible cultivar. So this was one of the significant findings, which gives us the idea that it's not only the RuBisCO Activase which is the real culprit, but RuBisCO also, they have different subunits, and then large subunit, it is highly heat labile as compared to the RuBisCO Activase. So once we cloned, once we characterized, then we thought, let us go for the isolation and purification of recombinant protein of RuBisCO. So we planned and we executed the heterologous expression of RuBisCO Activase. So here you can see we could able to purify a recombinant RuBisCO Activase protein of approx 42 kilodalton, and then this protein was further used for the antibody synthesis. So we could able to develop a polyclonal antibody of this mutant RuBisCO Activase, and then this antibody was used for the immunoblotting assay of different tissues of wheat at different durations and under differential heat stress. So here you can see the Western blot of this RuBisCO Activase, which was screened using the polyclonal RCA antibody, which was developed because of the recombinant protein which we synthesized in our lab. So here you can see the RCA as well as the RuBisCO protein accumulation in contrasting wheat cultivar under control and heat stress. And easily you can make out that under heat stress, the accumulation pattern of RuBisCO and RuBisCO Activase both increases under heat stress. Then further we thought that let us validate the interaction between RuBisCO and RuBisCO Activase under heat stress. So what is the effect of heat stress on the interaction between these two, or binding of RuBisCO and RuBisCO Activase? So that we did through dot blot analysis, and here you can see with the increase in the temperature, so the binding of RuBisCO and RuBisCO Activase, or rather I will say that interaction, so that weakened with the increase in the temperature. So that was very significant result, that the interaction is highly prone to heat stress, and then the protein accumulation increases with the increase in the temperature. So these two outcomes we could able to conclude because of immunoblot assay. So we did this accumulation pattern profiling in some of the selected wheat genotypes, which was shown under field condition, and then here if you see staggered showing was the method through which heat stress was given. So here you can see normal and delayed sowing has been written here. So we collected the sample, and then it was characterized for the accumulation pattern of this RCA, RuBisCO Activase. And here you can see we could able to conclude, or we could able to find out the best accumulator of RCA protein under stress. So that is SD2733, SD3090, NW2036, and then SD3043. So here easily you can make out that even under a delayed sowing, because under delayed sowing plant is exposed to heat stress under field condition. So we could able to identify some of these best accumulator of RCA protein. So RCA accumulation increases under delayed sow condition as compared to that of normal one. So it's not only the RCA, we also characterize the RuBisCO as I told. So we purchased this antibody from Sigma because it was not feasible to go for heterologous and then synthesize it, because this is very costly affair. So we purchased this polyclonal antibody which was specific to wheat from Sigma, and then we did accumulation pattern profiling at different stages of contrasting wheat cultivar, that is a vegetative stage, milky ripe stage, mealy ripe stage. Let me tell you, this milky and mealy ripe are basically sub-stages of grain filling. We also did under control and heat stress conditions. So here heat stress was about 38 degrees Celsius for 2 hours. And here you can see with the heat stress, the accumulation of this RuBisCO also increases. Even though this difference, so it's very difficult because we have to repeat a lot of time to get this result. The problem is, since RuBisCO is the most abundant enzyme, so it's very difficult to sometimes get these significant variations in the accumulation pattern of RuBisCO. And we also able to conclude that the percent reduction in RuBisCO under heat stress was observed less in thermotolerant cultivar as compared to that of thermosusceptible wheat genotype. So we did characterization with RuBisCO also, we did with RuBisCO Activase also. Then we plan to go for site-directed mutagenesis to manipulate the stability or the structure of recombinant or mutant RuBisCO activase itself. Our hypothesis was that if the stability or the structure, if the stability of the structure of RuBisCO activase increases, so definitely it will able to tolerate a little bit 1 or 2 degree more temperature, and that will have an effect on the efficiency or the activity of this RuBisCO activase or the conversion of inactive form of RuBisCO into active form. So this is one model which we use. That is, we designed a primer, mutant primer specific PCR amplification we did. We used Q5 side-directed mutagenesis kit, and then we designed some of the mutant primer, and then we use this for the amplification of the gene which we clone in our PMAL vector. So these are the primers which we designed. Now the question is how to identify the hot spot. As I told earlier, our hypothesis was that if RuBisCO activase is stable, so it can control the damaging effect of heat stress. So we started characterizing the RuBisCO activase reported in some of the hardy crops like Citeria italica, Gia maize, wheat, some of the like cactus and other plants. So we retrieved the RuBisCO activase sequence from hardy as well as susceptible plants, and then we did cluster W alignment. And using the Phi2 software, we could able to identify the hot spots of these particular sequence of RuBisCO activase, and the variations also we could able to explore. So based on the characterization, software-based characterization, we could able to pick up 18 hot spots which were predicted to play a very important role in the stability of this RuBisCO activase. So here, this is one slide where I have shown few, but then we could able to identify 18 different hot spots having a potential to influence not only the structure but the activity of RuBisCO activase also. So we picked one by one all these. Here you can see we designed the primers, then we amplified that particular amplicon, and further it was used for the heterologous expression analysis to isolate and purify the recombinant RCA protein. So once we got the recombinant RCA protein, mutant protein, that was used for the activity assay, and then we started characterizing those mutant proteins with a specific site which was replaced or substitute with another one. Now here you see the activity assay of RCA wild protein with the denatured one, and then some of the mutants which we developed. So we could able to identify one mutant, that is RCA138, which shows a tolerance up to 44 degrees Celsius. So that was one encouraging result which makes us to think that no, the hypothesis which we had given, so that is feasible. The only thing is that we have to incorporate this substitution, or we have to execute this substitution with a backup of bioinformatic characterization. So again, we initiated this, and then we could able to identify one mutant which showed very high activity even under heat stress. So during activity, when the enzyme was incubated at a temperature of 40 or 42 or 45 degrees Celsius, in that case also the decrease in the activity was very minimum as compared to the other mutant or as compared to the RuBisCO activase wild protein. So that was one proof of concept which we got, which says that if we go for the site-directed mutagenesis, and if we hit the right hot spot, so definitely there is every possibility that you can mask the effect of heat stress on this heat labile protein, that is RuBisCO activase. And if RuBisCO activase is present over there, so even under heat stress, so RuBisCO, even though the inactive form is more, so this RuBisCO activase will try its best to convert inactive form of RuBisCO into active form by changing the conformation, by removing these inhibitors or sugar phosphate moieties. And once the active site is open, so definitely the percent decrease in the photosynthetic rate, that will be minimum as compared to the control situation. So that was one outcome which we get, and then further we mobilize this in model plant, that is Arabidopsis. We validated our proof of concept in Arabidopsis, and we got very encouraging results. So I'm not going to share that because that is unpublished one. So we got very encouraging results. Now we are planning to target wheat, but the limitation is that in case of wheat, we don't have a standard protocol for the development of transgenic. So now we are exploring other novel tools recently developed. So we'll try to use this proof of concept to develop or to remodel a climate smart wheat crop. This is two publications which we got out of the work which I have presented here. So this was a quite encouraging one. And these are some of the other publications where we used our experiment results to justify, to validate, and to prove or to hypothesize some of the methods through which we can develop a climate smart wheat crop. So these are some of the outcomes. Coming on to road ahead, so it's not the end of the pathway. So even if we characterize the RuBisCO Activase or the RuBisCO, till date we don't know how many other members are there. Even though the pathway has been characterized, but when you go by the different papers which are coming nowadays, so a lot of novel members has been characterized, deciphered, and placed in these pathways. So every day we are getting one or the other member playing a diverse role in this pathway, like carbon assimilation or starch biosynthesis or the transportation. So there is a need to find out other members of this RuBisCO as well as RuBisCO Activase. So in order to enrich the genetic resources so that we can utilize this for the development of a plant or a plant for future. Coming on to the another one, that is the interaction. So till date, RuBisCO Activase and RuBisCO interaction has not much been characterized. So there is a need to characterize the interaction pattern of these two proteins, and not only these two different isoforms of these two proteins also that we have to characterize, not at one event point. It should be characterized at different stages of the wheat under differential heat stress in order to develop a model for this. Even there is a need to develop a thermostable RuBisCO Activase and RuBisCO enzyme, because as I told, it's not only the RuBisCO Activase which is heat labile, but even the last subunit of RuBisCO is also heat labile. So it is two-way friendship. So one is helping, but other also has a problem with a heat stress. So there is a need to design and develop a thermostable RuBisCO as well as RuBisCO Activase enzyme if really we want to stabilize this decrease in the photosynthetic rate, or really if we want to increase the fixation, carbon fixation rate. So we have to go by both the methods. Then there is every possibility that we can use this RuBisCO and RuBisCO Activase as a potential marker in wheat breeding programs. And last, as I told, the activity assay of RuBisCO as well as RuBisCO Activase is not an easy protocol. It is a radioactive labeling based method which is very tedious, and it takes almost 2 days to get the result. So there is a need to standardize and develop a chip based method, or maybe some colorimetric method through which easily we can identify or easily we can calculate the activity of these two enzymes. So this will give a pace to the whole research work, and then it will be easy to characterize, to remodel, and to develop a plan for future which will have an ability to sustain the heat stress. So with this, I think I have rest my case. I would like to acknowledge Director IARI, Joint Director Research IARI, all my team members, especially School Coordinator Dr. Viswanathan sir, and a special thanks to Honorable Vice Chancellor MPKB Rahuri, Naik sir, Organizing Secretary, Joint Organizing Secretary, coordinators, and all other members who are involved in this webinar program. And a special thanks to NHP CAST program, because of which it was feasible to present some of the significant findings in front of you. A special thanks to all the funding agencies who has given fund to execute these experiments which I presented. So with this brief, I would like to thank each one of you for attending and then being the part of this webinar program. Thank you. Thanks a lot.
Okay. Thank you very much, sir, for delivering a very enthusiastic and informative lecture on the unsung hero of carbon assimilatory pathway, unfolding the story of RuBisCO activities in wheat under terminal heat through Omics Approaches. Sir, your lecture will definitely be more useful to the students and young researchers who have joined this webinar. So there are some questions in chat box and some have raised hands. So we may not able to take all the questions, but with your permission, I would like to take few important questions. Okay. First, Dr. Mohit Bhatt from FAO has two questions. First one, does air pollution affect RuBisCO activities?
Yeah, please repeat. Hello.
Does air pollution affect RuBisCO activities?
So I think so we have not worked on this particular aspect, but then my concern says that because in Delhi also we did this experiment, and then we grown this crop in it's called Jambu also, and then I could not able to find much significant difference in the activity of RuBisCO as well as RuBisCO Activase grown at these two different places. So maybe my answer is so not significantly there will be difference in the activity because of this pollution.
Okay, sir. Thank you. And second question, is PEP carboxylase better than RuBisCO?
And your voice is not coming.
Hello. Huh, sir. Is PEP carboxylase better than RuBisCO?
No, in what sense? Because PEPCO is one of the key enzyme in C4 pathway, and then RuBisCO is one of the key in C3. So in what sense he is asking? So definitely C4 is better than the C3 if you talk in terms of climate change.
Okay. Thank you, sir. Then second, Dr. Radha Gupta from Gwalior has a question. What effect of cold stress in comparison to heat stress in wheat RuBisCO?
What effect of?
What effect of cold stress in comparison to heat stress in wheat RuBisCO?
Effect of cold stress. I think we have to see because heat stress that we have done. Cold stress, I have not found any paper where people they have given cold stress and they could able to analyze the activity. But then, as I told, since RuBisCO and RuBisCO Activase has a specific optimum temperature, and if a plant is exposed to cold stress, so definitely it will hinder the activity of these two enzymes. So effect will be more or less similar to as explained in case of heat stress.
Okay, thank you, sir. Then next, next question: which instrument or method is used to identify RuBisCO mutant present in wheat plant?
Okay, so there is no instrument which can identify. It is only the sequencing through which we have to validate whether the patch which we targeted for the substitution, so whether that substitution happens or not. So that you have to analyze through the sequencing only. There is no way out that directly you can see or visualize. So you have to sequence the amino acid also, you have to sequence the nucleotide also to see whether that change has incorporated in the sequence or not.
Okay, thank you, sir. And next, Mr. Muhammad has a question. He asked, have you done any work related to rice? And if yes, what is the relationship between lipin content and RuBisCO content in rice?
No, rice I have not worked, but I think evening we have a very good talk by Dr. Vishwanathan, and so Dr. Vishwanathan team members, they have worked exhaustively on RuBisCO Activase in rice.
Okay, sir. Then okay, sir. Please continue. Okay, thank you, sir. Then next, Dr. Muhammad again asks second question. Many enzymes can catalyze hundreds to thousands of molecules per second, but RuBisCO can only get through 2 to 5 cycles per second. Is it possible to play around with all the components of RuBisCO simultaneously by replying the genome of the bacteria chloroplast to the boost up crop production in future?
So I think this question is quite lengthy one, and you have to forward it to me.
Yes,
so
sir.
that
Yes, sir.
the answer.
Then next question, Anirban Roy asked question, if number of flower is less, does here RCA play any role or only during grain filling it works?
As I presented these two, so this is common logic that photosynthesis is not happening only in grain filling. So starting from the vegetative itself, so photosynthesis starts. So the enzyme, the only thing is we have different isoforms of this enzyme, which has temporal as well as spatial regulation. So maybe the isoform of RuBisCO Activase, which is present during pollination stage, that will be not present during the grain filling. It is because of this reason that during pollination, when the temperature is around 25 or 26 degrees Celsius, so that time we have a different activity of RuBisCO or RuBisCO Activase. But when you see the grain filling where temperature is around 38 or 39 degrees Celsius, that time also RuBisCO Activase works, but that isoform is different as compared to this one. I think I have shown one domain mapping slide also where I showed the variations in different genotypes, contrasting genotypes, and then the activity.
Okay, sir. Thank you, sir. The next, Dr. Ravindra Sadbhai from MBKV Rauri has one query. He asked whether only temperature factor reflect on RuBisCO activities or
No.
activation.
So temperature is one. So we did enzyme kinetics also. So even pH plays very significant role. Even the substrate concentration has a drastic effect on the activity of RuBisCO Activase. So temperature is one parameter, but other than this, different variables are also there which affect the activity of RuBisCO Activase. Even the light has drastic effect on the activity of RuBisCO Activase. So we also reported that if a light is very intense, it has a drastic effect on the activity of RuBisCO Activase. So activity will decrease.
Okay, sir. Thank you, sir. Next, Dr. Prashant Sende has a question. What is the effect of greenhouse gases on RuBisCO activities?
Hello.
Sir. Hello.
Yeah.
Dr. Prashant Sende has a question. What is the effect of greenhouse gases on RuBisCO activities?
See, so direct, I think, so literatures, you will not get where people, they have characterized direct effect of these greenhouse gases except the CO2. So people, they elevated the CO2 concentration in OTC chambers, and they could able to find out that after a certain limit, that is around 650 or 700 ppm, so it starts affecting the activity of the RuBisCO Activase. So it will drastically decrease once CO2 concentration goes beyond 700 ppm, something like that. But at 350 ppm or 400 ppm, so it has not much effect on the RuBisCO Activase per se.
Okay, thank you, sir. Then next, Dr. Preetam Sende has one query. Can you suggest any simple PCR-based test for screening the germplasm for RuBisCO and RCA genes presence in wheat germplasm?
Can you please repeat your question?
Yes, sir. Yes, sir. Can you suggest any simple PCR-based test for screening the germplasm for RuBisCO and RCA gene presence in wheat germplasm?
Yeah, so we tried this. So as I told earlier that we screened different genotypes of wheat, and then we could able to identify some of the genotypes where RuBisCO as well as RuBisCO Activase activities was very high. So we compare the sequence and we observed some variations, and then from the nucleotide we designed a stretch of primers, and we tried that in contrasting with genotypes. So there was significant difference, as I told. So easily you can use these primers, and through simple PCR you can easily, this will be the preliminary one, but easily you can make out which one is a tolerant or which one will behave as a susceptible. So easily you can do that.
Okay, thank you, sir. Then next, Mr. Anirab Roy has a question. How to decide duration of heat stress? And does only relative expression of RCA is sufficient for preliminary study?
See, the duration of heat stress, that you have to decide based on the pilot experiment, because there is no standard protocol where it's written that one has to give heat stress for this much temperature and then this much duration. So for all the labs who are working in this area, so you have to standardize on your own based on the crop, based on your objective through pilot experiment. That is first part. Second part, relative fold expression, no, because we are using cyber-based chemistry to characterize the expression of this RuBisCO Activase. I think you all might be knowing that cyber-based chemistry is an approx method. It will not give you the accurate quantification or absolute expression of the gene. So for absolute expression, you have to go for Techman-based method. But since number of samples will be more, so it's not feasible every time to go for Techman-based chemistry. So it is not accurate.
Okay, sir. Thank you. The next question.
Nandalal.
Yes, sir.
You can send some. Okay, one or two can continue.
Okay, sir. Okay. Thank you very much, Dr. Ranjeet Rajan, sir, for replying all the answering all the questions. So once again, thank you very much. For the information of all the participants, there are still few questions and queries in chat box. So we shall try to pass on those to the experts and convey you the response of experts through WhatsApp group or email. Okay.
Correct.
So thank you very much. Now I would like to request Dr. Prakash Lokhande, sir, Associate Professor of Department of Biochemistry and coordinator of this webinar, to propose vote of thanks.
Very good morning to everyone. I profusely thank Honorable Vice Chancellor Dr. K.P. Vishwanatha sir, Dean and Director of Instruction Dr. A.L. Farande sir, Director of Research Dr. S.R. Gadak sir, Convener and Principal Investigator CAST CSAW NPK Viravari Dr. S.B. Gorantiwar sir, for approving the organization of this webinar and constant source of inspiration and encouragement. Respected Associate Dean PGI Dr. P.N. Rasal sir, Co-Principal Investigator Dr. M.G. Shinde sir, Organizing Secretary Dr. R.M. Naik sir, Joint Organizing Secretary Dr. P.L. Kulwal sir, and all participants from national and international organizations. It is my proud privilege to propose a vote of thanks on this webinar. I, on the behalf of this university and organizing committee, I extend deep sense of gratitude to Dr. Ranjeet Ranjan Kumar sir, Senior Scientist, Division of Biochemistry, IARI, New Delhi, for accepting our invitation and for giving detailed talk regarding ribulose bisphosphate carboxylase oxygenase bifunctional enzyme in its enhancement or its maintaining its activity by use of RuBisCO Activase enzyme at proteomic and transcriptomic level to enhance the crop productivity under high temperature. Thank you, sir. Thank you very much, sir. I extend my sincere thanks to Dr. Prashant Kale, Dr. Nandalal Deshmukh, Engineer Mohsin Tamboli for untired effort to make the success of this session. I thank all the participant faculty, students from national and international organizations for actively participation in webinar. With the permission of organizing secretary and co-convener of the webinar, shall we close the sessions, sir?
No, it has got some announcement.
Sir, some I will announce some notices. Okay, thank you very much, Dr. Lokande sir. Before we end the session, we have few announcements. We have one more session in afternoon, so we will start the session at 4 p.m. And for this session, Dr. Vishwanathan Chinnaswamy, Head Division of Plant Physiology, IARI, New Delhi, will delivering the lecture on genome editing for enhancing climate resilience of crops. So all participants are requested to join afternoon at 15 minutes prior to schedule time.
Correct.
Immediately after end of the today's afternoon session, we will immediately start our validatory function of this webinar. So all participants need to be continue logging to the session till the validatory function is over. And we already invited you all for this validatory function on WhatsApp group. But again, dear participants, you are cordially invited for this online validatory function. And it is compulsory to all the participants be present at validatory function. And the attendance of today's evening session will be recorded at the end of the validatory function. So, Dr. Kulwal sir, any more announcement?
Well, thank you, Dr. Nandlal. Just post the PDF copy of that validatory session on all the groups so that they will understand. I think it has not yet been posted.
Yes, sir. Yes, sir. Oh, okay, sir.
Okay.
Thank you very much. With the permission from the organizing secretary of this webinar and speaker of today's session, we would like to close the session. So see you afternoon at 4 p.m. Thank you one and all.
Thank you. Thank you, Dr. Ranjeet Ranjan Kumar.
Yes, sir. Thank you.
Thank you.
Thank you, sir.